Imagine you're staring at a petri dish of glowing algae and wonder if those faint flashes of light are just bioluminescence, or could they be a secret language of cells, a phenomenon known as biophoton communication, where living molecules exchange ultra-weak, ultra-fast optical signals to coordinate their activities. What are biophotons, anyway? In 1923, Alexander Gurwitch first spotted faint ultraviolet glows from onion roots, and later research revealed that every cell emits a trickle of photons, perhaps just a few dozen per second, generated by excited biomolecules like flavins and reactive oxygen species. Question: can such feeble light really carry information? Answer: experiments show that cells synchronize growth rates or stress responses when linked by transparent fibers that only transmit their biophotons, suggesting these optical whispers influence distant neighbors. How might this quantum chatter work? Cells could modulate photon bursts in frequency or timing, akin to Morse code, using protein conformational changes as emitters and light-centric receptors like cryptochromes to detect signals. In plants, ultraweak blue-green flashes correlate with sun-tracking movements. In neurons, rapid photon pulses might accompany calcium waves, hinting at a parallel optical network beyond synapses. What examples have been observed? In one study, cultured cells separated by quartz filters, which block chemical diffusion but pass light, still coordinated division cycles, stopping when the filters were opaque. Another found that injured fish fins healed faster when nearby fins were separated by UV panelist barriers, implying long-range photon signals sparked regeneration. So could this redefine medicine and computing? If cells truly converse via light, we might harness biophotons to steer stem cell differentiation, detect cancerous communication breakdowns, or build bio-optical circuits that merge living tissue with quantum processors. Imagine implants that listen to your neurons' photon code and correct misfires in real time. Is it all too speculative? Skeptics note background thermal noise and the tiny photon counts challenge detection and fidelity. Yet, advances in single photon sensors and time-correlated measurements are revealing non-random patterns in biophoton emissions, patterns too structured to be mere noise. Why should curious beginners care? Because biophoton communication sits at the frontier where biology, quantum optics, and information theory collide. It suggests organisms may use nature's fastest messenger, light, to coordinate life's processes, opening a new window into how molecules talk at the speed of photons. Whether you're a biologist, physicist, or computer engineer, exploring these cellular light shows could spark breakthroughs in health, computing, and our fundamental understanding of life. Biophotons are weak ultraviolet to visible light quanta emitted spontaneously by cells and biomolecules during metabolic reactions involving reactive oxygen species and excited molecular states. These emissions occur at intensities of a few to thousands of photons per second per square centimeter, far below the sensitivity of our eyes, but detectable with sensitive photomultipliers. The hypothesis of biophoton communication suggests organisms might harness these ultra-weak emissions to transmit information over cellular distances. Investigating their spectral patterns and temporal dynamics forms the basis for understanding any potential signaling role. Plant roots, under stress, emit bursts of biophotons with distinct time signatures compared to unstressed controls. It's like tiny flickers from distant fireflies, individually faint, but potentially conveying Morse code-like signals if patterned correctly. Traditional intercellular communication relies on diffusible chemical messengers, hormones, neurotransmitters, and cytokines that bind receptors to trigger biochemical cascades. In contrast, photonic signaling would use photons as carriers, encoding information in pulse frequency, phase, or spectrum rather than molecular concentration gradients. Photons travel at light speed and are less subject to diffusion delays, offering theoretically faster communication. Distinguishing these modes highlights unique advantages and challenges of photon-based messaging in biology. Neurons fire chemical synapses in milliseconds, whereas a putative photon pulse could relay a signal across tissue in microseconds. It's like comparing mailing a letter to sending a laser pulse across a room. One is slow in bulk, the other is near instant and point-to-point. Key biophoton sources include oxidative reactions in mitochondria and peroxisomes, where reactive oxygen species generate electronically excited molecules like singlet oxygen or excited carbonyls. As these excited species return to their ground state, they release photons whose wavelengths reflect the specific chemical transitions. Cells can modulate ROS production and antioxidant defenses, potentially shaping the timing and intensity of photon bursts. Understanding these mechanistic origins is crucial to decoding any informational content. Mammalian fibroblasts exposed to UV light show a transient spike in singlet oxygen-generated biophotons lasting seconds. It's like sparks from a welding torch. Each spark's color and flash chemistry. Some researchers propose that biophotons exhibit partial coherence phase relationships over short time windows, analogous to laser light, which could enhance signal-to-noise in cellular environments. Coherent emission would allow cells to detect and distinguish photon patterns against thermal and biochemical background fluorescence. Quantum coherence effects, if sustained, might facilitate more reliable information transfer than purely random emissions. However, maintaining coherence in warm, aqueous conditions poses a major thermodynamic challenge. Low-temperature experiments show isolated chromophores can emit coherent photons, but in vivo evidence remains debated. It's like trying to hear a whispered song in a noisy cafe. Only coherent rhythmic tones stand out clearly against random chatter. Hypothesized pathways include intracellular waveguiding along microtubules or collagen fibers, directing photons between organelles or neighboring cells. Microtubules' refractive index contrast could channel photons internally, while extracellular matrix components form optical conduits across tissue. Such photonic highways might complement gap junction-based electrical and chemical coupling. Mapping these pathways could reveal an additional communication layer in multicellular coordination. Experiments show aligned collagen fibrils can guide blue-green light over millimeter distances with minimal attenuation. It's like fiber optic cables in a city's network, routing data packets around the metropolis faster than surface traffic. Biological tissues can exhibit nonlinear optical responses, second harmonic generation, or two-photon absorption that might amplify weak biophoton signals or convert their frequencies. These effects require specific molecular arrangements and incident intensities above threshold, potentially enabling selective enhancement of communication channels. Nonlinear interactions could also create frequency-doubled marker photons that unambiguously tag signaling events. Harnessing such phenomena would dramatically boost the viability of photonic messaging in cells. Collagen-rich tissues generate second harmonic signals under femtosecond laser excitation, demonstrating inherent frequency doubling capability. It's like a radio repeater that not only boosts a weak transmission, but also shifts it to a clearer frequency. If cells use biophoton signaling, engineered biosensors could decode physiological states via non-invasive light detection, offering real-time health monitoring. Photonic stimulation therapies might modulate cellular behavior, wound healing, neural repair by mimicking natural photon codes. Synthetic biology could design optogenetic modules to interface with intrinsic biophotonic networks. Realizing these applications depends on conclusively demonstrating functional photon-based communication. Early studies used photomultiplier tubes to detect cancer cell biophoton patterns distinct from healthy cells, hinting at diagnostic potential. It's like tuning a radio to pick up a station broadcasting health status signals from our own cells. Detecting ultra-weak biophotons requires extreme light shielding, low dark count detectors, and rigorous statistical analysis to rule out noise. Correlating photon emission patterns with specific signaling events demands simultaneous, high-resolution biochemical assays. Disentangling biologically purposeful signals from incidental oxidative byproducts remains contentious. Robust experimental controls and reproducibility are essential to validate any communication role. Replication efforts often struggle to distinguish true photon bursts from cosmic rays or electronic detector noise. It's like trying to record a whisper in a hurricane. Only the most sensitive equipment and careful filtering will capture the true voice. Advancements in quantum biology and stochastic thermodynamics may clarify the energetic and informational costs of biophoton communication. Computational models integrating biochemical kinetics with photon transport will predict feasible signaling regimes. New nano-optical probes, quantum dots, nanodiamond sensors could map photon pathways in living tissues. Ultimately, synthesizing theory and precise measurements will determine whether molecules and cells truly talk with quantum light. Simulations, coupling Ra's dynamics with microtubule waveguide models, predict optimal photon fluxes for intracellular messaging. It's like developing a grand unified theory of traffic flow that accounts for cars, bicycles, and pedestrians on a city's multi-layered network.